the viscosity. So the viscometer. So for some of the liquids and lapas and all etc. we will do with this. That is UV spectrophotometer you have also, isn't it? There we have. Yesterday we have gone through. So this is HPLC, it is Shimaju's HPLC. So it is see, it is a quaternary type where you can use 4 solvents at a time, and here this is an automated one. So here we will keep the samples, our extract what we have taken, no, that has to be kept here, and then we have to give a software how many we kept and what we are, so it will take. So otherwise manually there will be site where we have to, through the syringe we have to give that, we have to load it. So here it is there, then we have to start it, then we have here the column area is there. So column will be inside. HPLC. We have a column in here. So from here the extract will come, and here the solvent will come, and it will go through that. Then it will be separation will occur in chromatography. Then it will be coming here. Then here is a detector. Then it will go to the PDA detector, and then we will get the images here. So here we will be able to study the dissolution. Means there we have studied disintegration. Here we see the dissolution means in various, so gastric media, jejunal, duodenal, intestinal. When our drug goes, if it is in a tablet, whether how it dissolutes in it, and those can be studied. And if you have a membrane, you can see whether they are coming out. So various pH we can maintain, and it is having automation also. We can keep the multiple samples, and because when you do which has to, if you do in a company, you cannot do one by one. So you will do around 20 batches, 30 batches, and then will run on which is good deal again. That's it. Automation is also there. So AS. AS. Atomic absorption spectrophotometer. So this is from Shimazu. So in this now we can do heavy metal analysis or any elemental analysis, so potassium, calcium, zinc, copper, but you need the bulbs, the wavelength bulbs, the specific bulbs will be there for each. We need to have, and we need to have, here it is a furnace model, we need to have a graphite model for having mercury and others. You can add those things, and you can have the heavy metals. This is AS.
Qualitative.
Cryostat. Not cryostat, sorry, lyophilizer. So this is the lyophilizer. Lyophilizer means, so here we keep our, you have learned the extract method by either cold maceration or after you have to evaporate in water bath or whatever. But here by using the what we call freezing, so it will go to -40, then there is a vacuum, the moisture will be removed, and it can be made into a solid material. So you are getting that Maggi sachet and all those things. Their oil will be there. If you put it in a water bath, you can't get it. So here you can make them into powder. So this is lyophilizer technique. We have done some studies on this by instant basti. So basti has been made into lyophilizer, and we made it into powder. Then we can return to conjure, which this will work. So what will happen is, this is the place where we have to give sample. There we are putting with capillary or something, but we have, there is a syringe will be there. We have to put a syringe here, and we have to keep the TLC paper here, and then you have to load your drug extract. Then through the computer you have to tell what is the place it has to give band, what is the length and everything. You can decide from there, and then it will give, put the spots. It will move and put spots 1, 2, 3, 4, 5, 6, 7 like that. So then you can operate from here. You can tell 5 microliter, 10 microliter, how much you have to, how many times, everything can be determined here. After that, you have to run in a TLC chamber. So we don't have automated TLC chamber. We run through the normal chamber only. But you can have automated chamber also. But it's very costly. So usually people don't take. So chamber, we have to run in the same chamber, TLC chamber. Then after that, you have to, this is a hard plate, you can dry it. Then it will go to the next step. This is a one more thing is there. Here this is yesterday you might have seen this UV chamber where you can visualize it in normal light, that is white light, then you can visualize into short wave and long wave. So here you can visualize the TLC plate here, and then later this is a densitometer. There is one more will come, so that is called visualizer. So if you put it in visualizer, you can take a photographs what you will see, with the TLC paper with colors bands. Here we have to take with camera, but some problem will be there when you adjust your camera setting and all. But you cannot take in various wavelengths also. You can see only here. Visualizer will have whole wavelength 200 to 900. You can select and take. So there you can have a good in the whole range. After that, we will put that in. And then is to this is a TLC scanner. So we have to put our TLC plate here. Then this is a densitogram. Once you run it, what will happen is you will be getting like bands. That type of things will come. So where you have put spot, where band is there, you will get a peak. In TLC paper, you will be getting bands. Each band will show as a peak. So the length of the peak is its concentration. The where it is coming is qualitative nature. It will come at piperine will come at 0.4 or something. So it is depending upon the method. So you know that qualitatively based on the RF value, qualitatively, and quantitatively through this densitogram, because HPTLC, if this is not there, nothing is there. Because all TLC we do is same except this. For that we are using this to give exactly. I am putting 1 microgram in 1 ml. Then we are putting 5 microliter here. So 5 microliter is giving this much band. So you can estimate through software will be there. You can calculate through Excel also. You can estimate how much based on the peak length. So usually you calibrate. You take piperine 1 mg, 2 mg, 4 mg, 5 mg. Run it. You will get a straight line. Then you run your sample where it is coming. Dry a line. Then you can get it. So there are formulas, and so you can tell what your drug is. So quantitative purpose, this is very important. There are also one sonicator is there. So sonicator is for shaking the liquids. So this is by mixing. There, if you keep, there will be ultrasonic waves, and it will, then there is a oven. Then here we have autoclave, etc. Our Ayurvedic project is going on. Is that Russian? So he is working on Jivanti, DNA fingerprinting. So Jivanti various species they have collected. Then they are doing the DNA isolation. Then RFPD. Then they are also sending for genome. So this is how we have to run that, and then their DNA ladder will be there. We have to see the base pairs have come or not. So once you do so first you have to establish that your sample has enough DNA. Then you have to go for other things.
Sir, here we are using two of the instruments. One is regular PCR, thermal cycler, and another one is RT-PCR. That is where we are using reverse transcript enzyme to convert mRNA to cDNA and expression studies. That what we are using in COVID-19 detection, diagnostic one. Here, this instrument earlier been used for diagnostics, and later on, last 5 years to 6 years, we are doing some gene expression studies from different sample like medical, dental, physiotherapy, different genes we are looking for. And depend on the genes, that we cannot particularly tell about a particular disease. But if you are detecting any gene, expression can be done. Like last time we have done some isolation of fungal species and bacterial species for their enumeration, and then antimicrobial efficacy studies of nanoparticle natural product. We usually prepare from other laboratory, other PhD students they prepare, and they want to identify the effect on the bacteria. What we do is in microbiology we do MIC studies like using some different experiment like microdiscipline assay, agar diffusion assay. So that is what culture based method. But we are doing a PCR based method. Here we are going to check the virulence factors, whether your compound is going to act on particular virulence factor of the bacterial cell wall or fungal cell wall. So that also we can be able to check through PCR, and we had checked some of the activities based on efficacy studies. Like also we have done IBD work also. Like earlier, one of the faculty, Dr. Sanjay Mishra sir, was working on IBD, and later on also, like last few people, students, they work on IBD in irritable bowel disease, and related activities also we do. And sir, by collaboration from the Ayurvedic preparation also, sir, we have done. You have procured flow cytometer. It will help to enumerate. Enumerate means count the cell for in particular cell lines. We are using cell lines also. We to count the cell to identify the gene expression, and then cell cycle studies. Like we do cell cycle studies, like different stages of the cells, in what stage of how the cell is behaving, and number of cells and expression of particular gene can be done through flow cytometer, because we need to raise the antibodies, particular antigen, and that will be depending on the fluorescence intensity, it can be done. Like we are saying, sir, like in cancer, we are doing apoptosis and necrosis. Pair two of the maybe your compound may be apoptosis, apoptotic in nature, or necrotic in nature. So that we need to identify through some genes. Apoptotic gene markers are there, so that we need to go down the thing and select the apoptotic gene marker, and we need to feed that particular primer or particular gene marker, so that if in case that apoptotic marker is expressing in your cells or cell line, treated cell line, then it will show, otherwise it will not show. And we need to take, to compare that, we need to take a standard, like standard anti-cancer agents, like we are using doxorubicin, cisplatin, and different compounds we are using as a standard. So we need to compare the standard always to check the cell cycle. And the CDK protein that we express during the cell cycle, that protein will express. So that particular type of CDK, what type of CDK proteins are released by the cell, it can be detected, sir. And for that to detect, we need to generate the marker. Marker is very important, that what they are doing is different, same only, but applicable to plant. And marker related to cancer is also applicable to the cancer cell. So that marker means primer. Primer that will go and attach to the target gene and amplify the gene in a regular PCR. Here it will just show the quantitatively how much the gene is expressed in a flow cytometer. So like that we need to generate antibodies here. We need to generate here primers. There we need to generate the marker.
By taking which type of sample, blood or body fluid?
Yeah, any type of sample we can.
Blood
also we can take, body fluids also we can take, cell tissue samples we can take, embedded paraffin tissue sample of many years, embedded in the paraffin solution, so we can take. That is not the issue, sir. That we can able to take, and check the particular expression of the gene. It can be done. And compared to studies, neurological disorders also be linked, because stomach is directly linked to the brain health. Alzheimer's disease, and nowadays it has been proved that microbiome changes in the microbiome in the IBD, like in the stomach region, it will affect the nervous system. So Ayurveda has lot of implications, but we need to understand and scientifically validate that it is possible. Like we have done some commercial and that blood agar base is needed for the anaerobic bacterial isolation. So that we are using, it's all a normal technique, sir. Whatever I am showing here is a very normal technique used in every corner of the world. But that in our laboratory, what we are doing, some of the collaboration work we have done, and core area applied microbiology and cancer biology, where I am working in both the field, exploration of human gut microbiome and their importance. I told one of the work we did, and interdepartmental work we are doing for testing of the plant product that we most of the them we receive from different colleges, and Ayurvedic college is the one where students are coming for test the plant product. And these are some of the collaborators, sir. These has been we need to update it, and then research area, and then number of publications what we have generated. And till today, sir, some of last 5 years we want to do some 33 publications and cumulative impact of 63 or 64. And some book chapters have been published. And what I come to know is collaboration work gives us lot of ideas and lot of avenues rather than we are doing on one faculty doing in only one area. Like the collaborations work are very, very important. And if you have any questions, sir, like we able to answer. Like
3.11 software. Basically, it works on monochromata, which will have only one. It will detect multiple wavelengths. Okay, so we are going to perform 96 plate of assay. Only 96 plate of assay will be taken by this micro ELISA plate, not 48 or 360 well plate. Okay, so recently we have done so many assays, one which we have collaborated with Ayurveda College, where we have detected certain markers like TNF alpha, IL6 and leptin adiponectin. And other thing is we have done the COVID projects as well with collaboration with the public health department, where we have detected IgG antibodies and neutralizing antibodies. So what this will do, once you are done with your plate format, you have to place your plate into the micro reader, where it will take plate inside, and you have to set the plate format into the application. So then when you give a go to the application, it will take the plate inside and it will show you the absorbance rate based on the particular wavelength. So we have, at a time you can analyze 5 wavelength in this software at a time. 5 wavelength absorbance you can see. So whatever absorbance you want, you can set up and you can take the readings of that particular assay. And that is the micro plate reader, which is designed. This is fully automated, that is semi-automated one. This is fully automated and it will wash and dry the plate by itself. You just need to take out the plate and you need to dry. This is we need, because after each ELISA step you need to wash the plate, so the next step can be performed. So you need a micro plate washer. So this both work simultaneously. If you have any collaborative work you are planning, you can collab with us.
Beaker. Okay. You can see that there is some carbon particle you can see. It should not be there. Okay. It should be completely ash. It should be completely carbon free. Okay. Then what you have to do? You have to take 25 ml of 2 molar hydrochloric acid. How to prepare 2 molar hydrochloric acid? That's all they are given the appendix of pharmacopoeia. Okay. Then you have to take 25 ml of hydrochloric acid, 2 molar. If it, along with your drug, your ash, along with hydrochloric acid, if it effervescence comes, then one test is positive, there is carbonates are present. This will contain the carbonates, and it should not be like this black. It should be completely white. Then only you can call it as a ash. Okay. Then you have to boil it for 10 to 15 minutes in a water bath. Or it should be warm. At least you have to warm it, or you have to filter paper. It is made up of cellulose. Around 400 degrees Celsius, it becomes nil. The weight becomes nil. So that what common filter paper, what we are not using for the made, we are using Watson's filter paper. These are made up of the cellulose, and at 400 degrees Celsius, it becomes the paper weight becomes nil. So that only will collect whatever the insoluble matter. the paper weight will not get. It become a nil. Okay, then you have to filter it. First you have to moist with the distilled water. You have to moist the Is there a difference between one major difference is the measuring cylinder, this is the beaker. Other than this, you can feel any difference in both glasswares? Yes, madam. These are called as class A glasswares. These are called as class B glasswares or class 2 glasswares. Commonly, we are using this for quantification because these are accurately, these are calibrated in 27 degrees Celsius in room temperature. Exact 100 ml. Temperature also varies because ice, water, especially differ because temperature also plays very important role in the measurement. Okay, then we have to filter it. First we have to make the moist make. Then we have to filter it with the Wattsman filter paper. Take some precautions. Spillage should not be happen because you may get the error in the results. We may lose whatever our yield is, sand or whatever there is that insoluble matter. We may spillage may can give the error. So that precaution will be taken. The filtrate no worry, but the whatever our aim is, we are estimating the insoluble matter. That here to be taken. Then just you can see roughness. There is nothing but sand or other particle. That should be completely collected for repeated washings. Just you can feel that you can get the rough like sand. Protein, carbohydrate, plant acids, mucilages, tannins. Okay, this is for the inorganic elements. We have to prepare the ash even for the metal analysis like AS, arsenic, cadmium. It requires a ash. First, you have to make it ash, then you have to test it because we have to convert it to the inorganic form. It is clear filtrate, you can see. Okay, this is a filtrate. It contains the inorganic compounds, maybe sodium, potassium. That depends on the drug. They are given 3 ml. This is a qualitative test you can take. Then you have to add ammonium thiocyanate. Amruta? Or you can prepare the ammonium thiocyanate solution. Or directly you can add the crystals of thiocyanate. Pinch. Okay, just to add, if it gives blood red color, then it contains the iron. Iron is present. It's not like that much blood red color. Manjish chala hai to. Okay, this iron is present. Like that you can test. carbonate solution to test the calcium. Dropper kodu. No madam, drops. It should give a white PPT. Only appearance are coming. It is not. PPT. PPT is not there. Sometimes what happens? It requires some more test. It is only giving the appearance. It's not a PPT is not for. Already. It's test for sulfates. Test solution plus few drops of barium chloride solution. That too 5% of barium chloride solutions. It should give 5% barium chloride solution to give the white crystalline, and it should be insoluble in the hydrochloric acid. That is a confirmative test. No PPT. Sluggish. Okay. It is forming. Forming. Okay. You have to take test sample. Amrita, is it bare kodu dropper? Madam, chloride test. This is a lead acetate solution, 10% lead acetate solution of barium chloride. If it gives a PPT that contains a chloride. Few drops of sodium cobalt nitrate, it should give a yellow PPT. Its PPT it should be for sodium. Potassium. Test for potassium. No PPT. No PPT. Only color for PPT means it should like this. Curd like appearance should be there. You have to make a fold. You have to transfer it to the same crucible. Then you have to keep in the furnace for 15 to 30 minutes. 450 to 500 degrees Celsius. This is a crucible around more than 500 degrees Celsius temperature is there. Then we have to take it the crucible very carefully, and you should be always stand in the side of this furnace. Sometimes the cloth not from the cotton bale with chances of capturing the flame. Okay, and you should be always you should keep the muffle furnace around the window and around at the water easily available. Like you should not place in the even near to the furnace. You should not keep any flammable chemicals. You should take very care because the temperature is more. There is a chance of capturing the fire. Okay. Then we have to slowly remove it. We have to keep in a desiccator. Still it requires some time to make a the ash. Okay, then we have to keep in a desiccator and cool it. Filter paper. Then we are transferring into the furnace.
And it is active rotary operator. 3 things are there. So this is hot water bath. So here we put water And in this class we have taken our extract, which we are taking either the cold extraction or hot extraction. And we fill in this. This is the 1000 ml. Here you can see. So we can take up to half or 400 or something. So you need space because vapor will come and you are removing also. This must be liquid. So you should see how that is coming. So based on that you can take the quantity. Then this is easy. So you can remove the clamp. और इसको ऊपर करेंगे ना
ये पावर बंद करके इधर
नहीं बंद करना
पंप का भी बटन चालू है
See, we have taken extract and whenever we are applying, we have to apply a small quantity of glycerin here so that it will not liquid will be attaching here. So it will tightly binds. So you just fill it and close it and put the lid. So that when it is rotating, it will not go. So here you can, when you on it, you will be able to see this panel. So here you can set the rotation, how much. So you set it and put, then you rotate. This is rotate. Rotary operator, it rotates. When it rotates, what is happening? Then see the thing will come. The solvent is forming, the extract solvent is forming. So that also will be having same way. So evaporation is faster. So to keep on a water bath and keep, so only surface area is less, now surface area is increased. So that's why it will be faster than water. That is one thing. Why we are using it? Making very fast. And here water bath, here you have to set the temperature of the solvent. Here it is ethanol. So we are putting 55 to 65. So we kept 65. But showing you very fast it will come. You can keep 55. You can reduce also because we are using rotation, we are using vacuum, even 40 and 50 also we will do it. If you are very particular that volatile compound should not go, then we keep low temperature, we keep the vacuum, speed and all, but you take every quantities and you can run this your design. So you can set the rotation, how much faster the rotation, more the thing will be there, but faster the things, basically you should know how this liquid is behaving based on that. That you have to run and see and decide how much I have to give. So once we run it, it will be moving. Then this stand can come down. This should touch the water. So that's why here buttons are there, arrow buttons, down arrow, this is up arrow. So it should touch the water. So if you accidentally should not touch, then you push this button, it will go. When you here, it will not go down, so that it will not touch it. Already it will come up to this only, it will not go further down up to this only, but still sometimes this may break. That's why there is lock system. So when we put this, push it, it will be rotated. Here we have to this panel. We have to set the temperature. So I am increasing the temperature and decreasing the temperature. So I set it 65 and put a button, push. Then I should see. So it is having 44, now it is set to 55. So this shows that it is heating. Once the heating is there, then it will not show. So it is having the thermostat, we have set it, if temperature comes down, it automatically starts, if it's heating is more, it will start. So now it will heat and get to 55. After that, here once heating, vapor will come into this, it will be filled. So hot vapor, when it comes in contact with the cool water coming from this chiller, filled the water inside. So we have on this, so then we have set it to 4 degrees centigrade so that it will become cool. And there is a pump, when you on the pump here, the water will be coming from here lower and it will move and it will go there again. Cool water will be coming here because it is coming in hot, so it will become hot here, so that will go there and come back and cool. So we can maintain the coolness so that the vapor will condense and will be collected in this collecting flask. So once this is running, so we want to do extra, we apply vacuum here so that when more vacuum we are increasing, what is going to happen is it will take the negative pressure and the boiling will be faster. So you can see if we are increasing the vacuum, it will be very high. So it has to be controlled very meticulously. You should be there with your hand. Vacuum pressure increases, this will come into this. So if it comes into this, then your experiment is lost. Suppose you are doing quantitative purpose, your things are lost. Again, you have to redo it. Qualitative, you are doing then, but still you have to clean all these things and you have to restart and all. So that's why we should be very careful when we are doing this vacuum, and more vacuum also may break this. Second, as vapor comes, you have to maintain the temperature of the solvent, not more than that. More vapor will come, it will have heat and then more accumulation may cause flames. So you should see that it is working, it is cool and not high speed, high temperature is there, so that more flames are coming.